Hey class, welcome back. We're going to talk about RNA and protein synthesis. You may notice I am in the same outfit, in the same seat, same hair, same look. Well, it's because I'm still at home and I'm just recording these lectures right in a row to get them all done and ready for you. All right, so let's get going. We just spoke about transcription last lecture. Transcription was a process of taking the DNA message and copying it into messenger RNA. So translation is going to be taking that message once it leaves the nucleus, goes out into the cytoplasm, and making it into a protein. It is the decoding of the mRNA message into a polypeptide chain, which is another word for a protein, meaning just many peptide bonds in a row. So this green guy with the holes in it right here, that's the nucleus. Your long single-stranded chain here of nucleotides is your messenger RNA. And here you have your ribosome, which is your rRNA. Your ribosome is composed of a smaller subunit on the bottom and a larger subunit up top. All translation takes place at ribosomes. Either the ribosomes are free-floating or connected to the endoplasmic reticulum. And during translation, the cell uses information from the messenger RNA to produce the proteins. The messenger RNA tells them which amino acids to link up in which order. But before the process can occur, there's some kind of cleanup that we need to do of the messenger RNA. So the messenger RNA, when it is made, it is just an exact copy of everything that's in the DNA, but not all the information is necessary for making the protein. So there are specific enzymes that'll cut out chunks of information because they interrupt the coding sequence. So introns are what we call the pieces of information that get cut out because they interrupt. So introns interrupt, so introns are out. Exons are what are left behind to be expressed or translated as needed. So you will end up with a shorter sequence as those pieces are cut out. The introns are cut out for interrupting, and the exons are left behind and spliced together to be expressed. This is another picture of that same process with the original strand in blue containing both the exons and the introns. Then the introns being snipped out and the mature messenger RNA being just the exons all spliced together. And now we're ready to make a protein. Protein synthesis. It is a fancy way of saying we're going to make a protein. To synthesize something is to make something. We've used that word throughout the course many times. Bases in your messenger RNA code for specific amino acids, which will make a functioning protein. So a group of 3 sequential bases on the mRNA strand is called a codon. So here on your right, you have a picture of your mRNA strand. And every 3 bases is a codon. So here the first codon is GCU, then ACG, GAG, CUU, CGG, AGC, UAG. I highly recommend, if you're doing any sort of translating or anything, making a little line between each of your codons to separate them out so you don't get lost within the strand and move over wrong one letter and put in the wrong amino acid. So that is something that I do to help myself stay in track with translating an amino acid sequence that has been given to me on paper. So the genetic code contains 64 codons, and that's every possible 3-letter combination of the 4 bases A, U, C, and G. And they code for only 20 different amino acids, including your start and stop signals as well. And what that means is that there are multiple codons that code for the same protein. or same amino acid, sorry. There are multiple codons that code for the same amino acid. There are 3 different codons that signal the production of the protein to stop, that we call stop codons, and there's only 1 codon that signals it to start. Methionine is the amino acid, and the codon is AUG. It's at the beginning of every single protein. So if this is your mRNA strand, and you have your codon wheel here, the codon wheel is read from the inside out. So GCA would be your first codon, and you would read the G that is in the very inner circle first, the biggest one. So you know that your answer has to be in the upper left quadrant of this chart. Then you go to the second row, the C that is connected to that G. And then go to the next A in the 3rd row. And that brings you to the amino acid alanine, looks like to me. Sorry, on my screen it's super tiny, but I believe it's amino acid alanine. Like I said before, if you put a line between each of the codons, it will help you stay on track. And your amino acids are alanine, then threonine, then leucine, leucine again, and leucine again. You'll notice that there are 3 different codons here for leucine. So like I said before, there are many codons that will code for the same amino acid. That means if you have a substitution of an A for a G, but it's in CUA and change it to CUG, it's still going to make leucine. It's not going to be a big deal at all. The protein hasn't changed structure even 1 bit with that mutation there. So that would be a silent mutation. So the process of translation is the process of taking the messenger RNA unit or message and assembling your amino acids to make your protein. The first step is that the messenger RNA moves out of the nucleus and attaches to a ribosome. It feeds through the ribosome like a dollar bill through a vending machine. The transfer RNA transports amino acids into the ribosomes, and the anticodon on the tRNA, which is the 3 bases that are on the tRNA, bond to the complementary codon on the messenger RNA. Amino acids form peptide bonds and form a strand called a polypeptide. Poly meaning many, peptide referencing the fact that they're forming peptide bonds. So many peptide bonds is referring to a string of amino acids, which is making a protein. The stop codon on mRNA ends the process and the new protein is released. So there are 3 different stop codons, and if you look at the wheel, you will see them. And they are not an additional amino acid. There is no additional amino acid that gets put on the end. It is just what tells the entire process to stop. And the ribosome will release the messenger RNA. Everything will kind of break apart, and the process of creating the protein will be completed for that mRNA sequence. As the ribosome binds new tRNA molecules and amino acids as it moves along the mRNA. The ribosome binds new tRNA molecules and amino acids as it moves along the mRNA. This is a picture of the ribosome moving along the messenger RNA, and you can see the transfer RNAs in it. There are 2 already inside, one with the amino acid methionine, and then the next with the amino acid phenolphthalein. And lysine is on its way to bind to the next codon. In the next thing, you will see that the first one is breaking off, but it is left behind its amino acid. And the ribosome is just moving down the strand, reading the next 3, then the next 3. And as it goes, new tRNAs will keep coming in, bringing new amino acids, and the old ones will keep leaving to the left, leaving their amino acids behind. The polypeptide train comes out of the top of the ribosome here. And once it gets to the stop signal or the stop codon, everything breaks apart. So example UGA is a stop codon, so that is going to end the sequence of events. So the process of going from DNA to messenger RNA is transcription, and messenger RNA to protein is translation. And this is a picture of both of them. You have your DNA strand at the very top, then you have your messenger RNA strand, and then you have the corresponding polypeptide chain of amino acids, which makes your protein. A says that the messenger RNA is transcribed in the nucleus, so transcription occurs inside of the nucleus. And then it will come out through the pore, find a ribosome, and the transfer RNA for letter B will bring the amino acids and bind the matching anticodon to the codon. C, here's a picture of the polypeptide assembly line as the ribosome keeps riding down the messenger RNA strand, reading codon after codon and assembling the amino acids in a chain. The polypeptide chain is shown as in D, and as it is fully completed, and then once it gets to the stop codon, everything breaks apart. If you were to be a ribosome, you would need a codon wheel. So print out the notes. There is a codon wheel on there. And take a minute to translate the codons provided, or you can Google an example of a codon wheel. There are also codon data tables that work just the same way. You have your first codon, second codon, third codon, or first letter, second letter, third letter of the codon, tell you which amino acid it is. Go ahead and take a moment to translate this message. Hopefully you got methionine, histidine, serine, and leucine. If you would like to watch a video of protein synthesis in action and you are one of my students, download the PowerPoint, click on the protein synthesis video link, and you will have access to that animation. Thank you very much, and I will see you in class.